Hello and welcome. Today we are stepping into the shoes of a forensic investigator. We're going to dive deep into the science that solves crimes, where the biggest clue might just be a tiny, almost invisible stain. We're going to break down exactly how forensic biology turns that one little spot into a story, and maybe even a name.
But hey, before we get our hands dirty, if you like what we're doing here at Smart Tutor, do us a quick favor and hit that like button and subscribe. That way you won't miss out on our next deep dive. All right, let's head to the crime scene.
So, picture this. You've just arrived on scene. The air's tense, everybody's looking for answers, and you spot it, right there on the floor, a single, suspicious stain. Is it blood? Something else entirely? Right now, it's a total mystery. But that little stain, that's our starting point. It's our job to figure out its story.
And that's exactly what forensic biology is all about. You can think of it as a two-part mission. First, we have to answer the what question. What is this stuff? Is it blood, semen, saliva? We have to identify the biological material first. Only then can we move on to the second, and arguably most important question, whose is it?
Okay, so here's our game plan. We'll kick things off with serology, which is our initial screening. Then, we'll zoom in on how we identify specific clues like semen and blood. After that, we'll get to the real powerhouse, DNA analysis. And finally, we'll wrap it all up and review what we've uncovered.
So our investigation kicks off with serology. You can think of this as the triage of forensic science. Look, we can't just jump straight to a DNA test on every single stain we find. That would be incredibly expensive and time-consuming. First, we need to figure out a much more basic question: what are we even dealing with here? And that is the job of serology.
Now, to do this job properly, you have to know your tools. And for any of you studying this stuff, this part is absolutely crucial. We're going to be talking about two main types of tests: presumptive tests, which are like our initial hint, and confirmatory tests, which give us the final answer. Understanding the difference, the good, the bad, the why, is everything.
So let's break this down. This two-step process is really the heart of serology. First up, you've got your presumptive tests. Think of these as a detective's hot tip. They're super fast, really sensitive, and they'll tell you if a substance might be present. They point you in the right direction. But a maybe isn't enough for court, right? That's when you bring in the confirmatory test. This one is more like getting a perfect fingerprint match. It's slower, but it's highly specific and tells you with certainty exactly what you have.
All right, now that we know the difference between screening and confirming, let's actually put it into practice. We'll start with one possibility for our stain. In many cases, especially sexual assault investigations, identifying semen is absolutely critical. And our analysis is actually a two-pronged attack. We look for the seminal fluid itself, and we look for the presence of sperm cells.
Now you might be thinking, hold on, why look for both? If you find sperm, you found semen, case closed. And you'd think so, but it's actually not that simple. The reality is, some men don't produce sperm. This can be due to a vasectomy, certain diseases, or congenital conditions. They can still produce seminal fluid, but it might contain very few or even zero sperm cells. So you can see why if we only looked for sperm, we could miss a huge piece of evidence.
So here's our menu of options. For the presumptive or screening stage, the big one is the acid phosphatase test. Then for the conclusive, confirmatory stage, we've got methods that leave zero doubt. We can literally look for the unique shape of sperm cells under a microscope, or we can test for a specific protein that is only found in semen and nowhere else.
Let's really zoom in on that presumptive test, the brentamine spot test, also known as the AP test. This test is super clever. It works by targeting an enzyme called acid phosphatase. Now, the prostate gland packs seminal fluid with a massive amount of this enzyme. We're talking incredibly high concentrations, way higher than you'd find in any other bodily fluid. This makes it a perfect, glowing target for a screening test.
But, and this is a big but, here's the catch. This is why it's only a presumptive test. Other body fluids, like vaginal secretions, for example, also contain some acid phosphatase, just in teeny tiny amounts. Semen has hundreds, sometimes thousands of times more. The test is designed to give a super fast, really intense color change when it sees that huge quantity. A slow or weak reaction, that could be a false positive, and that's exactly why we always have to follow up with a confirmatory test.
Okay, so we've run our screen for semen. Now let's pivot and look at the other major possibility for our mystery stain: blood. I mean, blood is probably the single most common type of biological evidence found at crime scenes, so knowing how to identify it is just fundamental to forensic science.
So, what's the secret behind these presumptive blood tests? Well, it's all about hemoglobin, the protein that makes our blood red. There's a part of it called the heme group. Its main job is carrying oxygen, but it has this awesome side effect. It acts like a chemical booster or a catalyst for a specific type of reaction called oxidation. Our tests are designed to look for that supercharged reaction. If we add our chemicals and see that reaction happen almost instantly, we have a strong indication that heme is present.
And that one core principle is the engine behind a whole bunch of different tests. Phenolphthalein is the real workhorse in most labs. But you've probably heard of the most famous one from TV shows, luminol. That's the one that creates that eerie blue glow, perfect for finding cleaned up bloodstains. They all work on that same principle of heme boosting a reaction. They just give us a different visual cue.
This table is fantastic because it lays it all out for us. Let's take a look. You see phenolphthalein here, a presumptive test, noted with a P. It's based on that peroxidase activity and gives you a red color. Now, check out luminol. Same idea, but look at that sensitivity. It can detect 1 part of blood in 5 million parts of another liquid. That's incredible. Then on the other side, you see our confirmatory tests, marked with CN, like the Takayama test. And notice the basis is totally different. It's not about a color change, it's about forming specific crystals. That's way more specific.
Okay, so let's say our phenolphthalein test turned bright pink. That's a strong hint we're looking at blood. But a hint is not proof. We need to be 100% certain. And for that, it's time to bring in the big guns, the microcrystalline tests.
A classic choice for confirmation is the Takayama test. What happens here is we add specific chemicals to our sample. If blood is there, the chemicals react with the heme and form a brand new compound. And here's the beautiful part. The reason it's confirmatory, this new compound forms very specific pink needle-shaped crystals. Lots of things can cause a color change, but forming a unique crystal structure, that's like a chemical fingerprint. Seeing those little pink needles under the microscope is our absolute positive confirmation. Our stain is blood.
And there we go. We did it. Through serology, we've gone from a mystery stain to a positive ID for human blood. We have officially answered the what question. But of course, that just blows the door wide open for the biggest question of all, whose blood is it? And to answer that, well, we leave the world of chemical spot tests behind and enter the incredibly powerful world of DNA.
To get from a blood stain to a person's name, we have to understand the entire DNA workflow. How do you even get the DNA out of the cells? How do you read it? What are the different types of analysis you can use? And just as importantly, we have to be aware that even this amazing technology has its limitations.
The whole process really breaks down into 3 key stages. Step 1 is extraction. This is where we literally break open the cells and pull out just the DNA. Step 2 is quantification. This is a critical quality control step. We need to know exactly how much DNA we have to make sure the next part of the process works correctly. And finally, step 3 is application. This is where the magic happens, and we use specialized techniques to analyze the DNA and generate that unique profile. And during that final application stage, scientists have a whole toolkit at their disposal. The gold standard is STR analysis. This creates a unique string of numbers that's almost like a genetic fingerprint. Now, if you have a complex sample with DNA from multiple men, you can use YSTR analysis, which focuses only on the male Y chromosome to help sort them out. And for really old or degraded evidence, like an old bone or a hair without a root, you can use mtDNA, which is a tougher type of DNA that survives for longer. It's all about picking the right tool for the evidence you have.
And just like that, we have a full DNA profile from our bloodstain, ready to be compared to a suspect or searched against a database. Our whole investigation, which started with nothing more than a simple, unknown spot on the floor, has now produced one of the most powerful pieces of evidence imaginable. Let's do a quick recap on how we got here.
So, what are the big lessons from our case? Well, we've seen that forensic biology is a logical 2-step process. Serology first, then DNA. We learned that presumptive tests are our fast and sensitive screeners, while confirmatory tests give us that specific positive ID. We saw how it's all based on cool chemical principles, from heme's catalytic power to the formation of those unique crystals. And finally, we saw how DNA analysis gives us the ultimate answer, who that sample belongs to.
We've just seen how incredibly powerful DNA can be, pointing the finger at a suspect with mind-boggling accuracy. But that brings us to one last, really important question to think about. Is it perfect? Is it infallible? What happens when you have a tiny amount of DNA or a complex mixture from 4 different people? Or what if someone's DNA was just transferred to the scene innocently? Understanding the limitations of DNA evidence is just as important as appreciating its power.
And on that thought, our investigation is closed. We followed the evidence right from the beginning, from an unknown stain all the way to a potential suspect. Thanks so much for joining us on this one. As always, stay curious, and we'll see you in the next explainer.